Hello everyone, welcome back to the session. So in today's class we are going to discuss about the very shorts in the locomotion, reproduction and protozoa. Okay, so this is in the first year. Okay, so our first question will be draw a labeled diagram of TS of a flagellum. Okay, so here they are asking about the diagram and you have to draw a diagram definitely, and not only you have to draw a picture but also you have to mark a parts clearly. Okay, so that's what draw a labeled diagram of TS of a flagellum. Okay, see, so this is called as an outer sheath. Okay, so first you have to draw like an outer, draw a circle. Okay, and it has to be, it will be a double membrane and that is called as an outer sheath. Okay, so and inside an outer inside the flagellum we can see a round like structure, so which is nothing but a tubule. Okay, an alpha tubule and beta tubule. See, tubule is nothing but this is the subunit, this is the motor proteins of microtubule. Okay, so you can think that that will be the subunit of the microtubule. Okay, so microtubule is the one involving in the movement of the flagella. Because of the movement of the microtubule, the entire flagella will move. Okay, so that is why we are saying flagella will be the locomotory organelle. But how the flagella will move? Because of the presence of the microtubule. And this beta tubule and the alpha tubule, which is nothing but the subunit of the microtubule. Okay, and another important thing will be the dynanomes. Dynanomes is again, it is a protein, and this circle is the inner sheath. Okay, so this is considered as an inner sheath, and we are having central singlets. See here, all the flagellum will be in a doublet. Okay, so 1 2 3 4 5 6 7 8 9. Okay, so totally 9. Okay, 9 will be there, 9 doublet will be there, plus inside there is 2 singlet will be there. That is what we can tell that as a 9 plus 2 array. Array means arrangement. Okay, 9 means the outer doublets. Okay, we are having 1 2 3 4 5 6 7 8 9. Okay, so 9 doublet will be there. That inner, this is called as an inner central singlets. Okay, here the microtubule will be present in a singlet, not in a pair. So that is why 9 plus 2 arrangement, right? And there is a spoke will be present here, that is called as a radial spokes. Okay, and this doublets is called as a peripheral doublets. And besides the dynanomes, we are also having the nexin arms. Okay, so all these were the parts of the flagellum. Okay, so this will be the first question. And list any two differences between the flagellum and the cilium. Okay, see here, there is a flagella and cilia. Both are considered as a locomotory organelle, no doubt in that. So this flagella is a locomotory organelle, and what will be the structure of the flagellum? See here, it will be long, but the cilia will be short. Okay, and it is like this. Okay, it is whip-like and it is hair-like. So hair-like in the sense, so this is considered as a cilia, which is hair-like structure which is present in the outer layer of the body or any other part of the organ. Okay, and the functions of the flagellum will be locomotion only. Okay, see, you should you very clearly understood the function of the flagellum and cilia. Okay, see, the flagellum has only one function and that will be the locomotion only. Okay, see, cilia are doing the function not only with respect to the locomotion but also the food collection, sensory roles. Okay, so all these things have been done by the ciliary structure. Okay, and movement, the flagellar movement will be the undular movement. Okay, and the ciliary movement will be the pendular movement. So these are all the differences between the flagellum and the cilia. So if any question asks, like if list out any two differences, means you should not write only two. Okay, so you just add an extra point. Okay, okay, so this is the differences. Now the third question will be, what are the dynanomes and what is their significance? Just now we have studied dynan is nothing but a protein. Okay, and here the question is asking, why, what is dynanomes and what is their importance? Okay, see, dynanomes, it is a pair domes present on the A tubule of each peripheral doublet. So we are having 9 peripheral doublet. So in that doublet there is a paired arms will be there, that to specifically in the A tubule. So we have studied A tubule and the B tubule, and this dinonorms will be present on the A tubule in a paired form. Okay, so it is considered as a paired arms in an A tubule of each peripheral doublet, right? Now, what will be their significance? Enabling the bending movement of the flagellum by sliding microtubules past each other. Okay, so why the dinonorms has been present in the A tubule means, so this is the one enabling the movement of the flagellum by sliding the microtubule. So what I have mentioned you before, so the flagella is a locomotory organelle. Because of the flagella, the organisms can able to move. Okay, this is what we know that. Okay, but what will be the thing which is supporting the flagella to move? That will be the microtubule. Okay, and for that microtubule, who is exactly making the microtubule to slide past with each other? That is nothing but the dinonorms. Okay, dinonorms helping the microtubules to slide past with each other, and as a result of this movement of the microtubules, the entire flagella will move, and this movement of the flagella will make the entire organisms to move. So okay, this is what an ultimate thing. And what is kinety? Kinety means a longitudinal row of kinetosomes together with a kinetodesmata forms a kinety. See here, there is a kinetosomes. There is a structure which means this is considered as a basal body, okay, through which the flagella arises. Okay, where exactly the flagella arises is called as a kinety. Okay, and how can we define kinety? It is a longitudinal row of kinetosomes. Okay, a group of kinetosomes together with the kinetodesmata. So kinetosomes plus kinetodesmata together we are calling them as the kinety. Okay, now what will be the differences between the synchronous and metasynchronous movement? Okay, see, synchronous movement means here the cilia occurs in a transverse row. Say for an example, consider this is an organism, Paramecium. This Paramecium, they are having a cilia which is covering their entire body, and the transverse movement in the sense, so this is what a transverse movement, and this is was a longitudinal movement. Okay, so there is a different. Longitudinal movement is this, transverse movement is across the organism. So that is what the cilia is doing. They are having both synchronous movement as well as the metasynchronous movement. Okay, see here, cilia in a transverse row beat simultaneously in one direction. Okay, so those cilia, if it is present in the transverse row, they are beating simultaneously, so that will be in only one direction. So that is called as a synchronous movement, which means all the cilia beat simultaneously in same direction. That leads to a synchronous movement. Okay, that leads the entire organisms to move only in one direction. And the metachronous movement, so here the cilia is in the longitudinal row. So the cilia which is present in the longitudinal row, if they beat simultaneously, or if they beat sequentially one after the other, which means one cilia will move, and after the movement of one cilia, another cilia will beat, and followed by another cilia will beat. So this is sequential manner, one after the other, not simultaneously. Okay, if the transverse row of the cilia beats simultaneously in one direction, in the same direction, that leads to the synchronous movement. If the cilia, if it is present in the longitudinal row, they are beating sequentially, which means one after the other cilia, it is beating. So it leads to the metachronous movement. Okay, so now what do we refer to the offsprings formed by the asexual method of the reproduction, that is a clone. Okay, see here, they are asking what is clone. Okay, so clone, so whenever we are saying that as a clone, this is an organism, and the clone of this organism is this. Okay, so organism 1 and organism 2 is this. Okay, so as organism 2 will be the clone of organism 1, so how these two organisms will look like? They look alike, they look similar. Okay, so that is what a clone is, a group of morphologically and genetically identical individuals. So the clone is not only morphologically identical, morphological in the sense outward appearance, that is a physical feature. Okay, so the clone is a group of morphologically and also the genetically identical individuals. Okay, it is not similar in their morphological form, that is in the phenotype, it is also genetically similar, and they are formed from a single parent, and that too via the asexual reproduction. Okay, so if it is a sexual reproduction, there will be the involvement of two individuals. As a result, the offsprings will be look like both the organisms features has been present in the offsprings. Okay, so that is why it is in the asexual reproduction. Asexual reproduction means, so this organism is undergoing an asexual reproduction, they are producing an offspring, and now there will be no involvement of other organism during mating process. So you should know the offsprings will look like exactly as the single parent. So this is what clone. So the exact keyword here will be a clone, which is morphologically as well as genetically identical, and that will be from only one parent, not with the involvement of two parents, that too asexual reproduction. If there will be only one parent, definitely that will be the asexual reproduction. Okay, now what will be the distinguish between the proter and ophist? Okay, see here, I think we have discussed this term already. I think proter means anterior daughter cell in the paramecium binary fission. See, say for an example, this is this is a paramecium. Okay, so this is a paramecium during binary fission. See, just see here. So this is a paramecium. Okay, so this is a paramecium. So the transverse movement occurs. This leads to an anterior region and the posterior region. Okay, the anterior region will be one organism and the posterior region will be the one organism. Okay, and this will be the daughter cell of this parent paramecium. Now the anterior daughter cell, so that is what here, the anterior daughter cell is called as a proter, and the posterior daughter cell is called as an ophist. Okay, and here the proter, that is the anterior daughter cell, will receive the contractile vacuole, cytopharynx, cytostome from the parent individual, and the ophist, that is the posterior daughter cell, will receive the contractile vacuole only, which is present in the posterior side of the parent. Okay, then how this anterior proter will develop into a paramecium, how this ophis will develop into a paramecium. So what are all the organism which is not present in the daughter cells will be synthesized by themselves later. Okay, but here immediately after the binary fission, so these are all the organisms which is present in the anterior paramecium, that is called as a proter, and the posterior daughter cell called the ophis. Okay, so this is what the difference between proter and the ophis. Okay, next, the how is asexual reproduction advantageous in evolution? So we have two different types of reproduction, that is sexual reproduction and asexual reproduction, and so we are saying that in an evolution, definitely sexual reproduction will be advantageous when compared to an asexual reproduction. Okay, so why? See, in sexual reproduction there will be the involvement of two individuals. Okay, so if that will be, there will be, so this mother having a different genetic setup, father is having the different genetic setup. As a result, this offspring have a genetic diverse offsprings. Okay, so this offspring is considered as genetically diverse. So that is what this offspring is very advantageous when compared to the daughter cell of the parent cell, which is involving an asexual reproduction. Okay, and this leads to origin of new species over time. Okay, so over the period of time, this leads to an origin of new species as they are having genetic variation within it. Okay, now distinguish between lobopodium and phylopodium. Give an example to each. So you should know what will be the difference between lobopodia and philopodia. But before that, you should know both are all the types of the pseudopodia. Okay, so pseudopodia will be the locomotory organelle which is present in case of the amoeba. Okay, and based on the size and based on the shape, okay, based on the shape we can classify pseudopodia into 4 or 5 different types, and that two type will be of these 5 types, one of the two types will be this lobopodium and phylopodium. Okay, so the lobopodium will be blunt finger like pseudopodia. It's look like a lobes. The pseudopodia in lobopodium looks like a lobes. Okay, and the example, the finger like pseudopodia is called as a lobopodium, and it will be present in the amoeba and the entamoeba. Okay, now phylopodium, so the phylopodium will be the fiber like pseudopodia. It is present in the Euglena. So these based on the structure, pseudopodia will be categorized into five types. Okay, so that is what the lobopodium, phylopodium, reticulopodium, axopodium, so all these things. Now define conjugation with reference to the ciliates. Give two examples. So they have asking that define conjugation. So conjugation means, see, this is a bacteria and this is also another bacteria. There is an important structure called as a pili. Okay, so the pili. So this is the one is responsible for attaching two different bacteria just for the reproduction process. Okay, that is what a sexual reproduction. Okay, see, this is one bacteria or any other organism. Okay, so this is one organism and this is one organism. There will be the temporary union between two senile ciliates of different mating types for exchange and reorganization of nuclear material. So why these two individuals has to join together? So just to exchange their genetic material. Okay, so that is what we can say that as a sexual reproduction. Okay, the example will be Paramecium and Orticella. Just know this conjugation will be the attachment of two different individuals temporarily. It is not a permanent union, just a temporary union until up to the genetic material has been exchanged. Okay, so this can be seen in Paramecium and Orticella. Okay, so that's all about the shorts in that chapter, and that is the locomotion, reproduction, protozoa. And now we are going to discuss about the very shorts in the musculo-skeletal system. Okay, this is in the second year. Okay, so the first question will be, what is a motor unit with respect to the muscle and the nerve? Okay, see, the motor unit, this is a unit which is consist of a motor neuron. That is why the unit is called as a motor unit. Okay, and all the muscle fibers in it innervates. It is a basic functional unit for muscular contraction. See, we have said that cell is a structural and functional unit of life. Nephron is the structural and functional unit of kidney. Okay, neuron will be the structural and functional unit of brain. Okay, so that is why we are saying there is a motor neuron is there, and this motor neuron is present. Motor unit consists of a motor neuron. Okay, and it is present in the muscle. Okay, because for muscle cells, this motor neuron is the one sensing the signals. If I am going to touch here, who is going to sense the touch? Okay, motor neuron. So that is why in the muscle cell, the muscle contraction, the muscle fibers, the presence of motor neuron is very important. Okay, now what is the triad system? So triad system, in the sense, this refers to the structure in the skeletal muscle fibers formed by one T-tubule flanked by two terminal cisternae of the sarcoplasmic reticulum. It helps in the rapid transmission of impulses of muscle contraction. Okay, see, they have asking the triad system. So what is meant by triad system? So this is the structure which is present in the skeletal muscle fibers. Okay, so this triad system will be present in the skeletal muscle fibers. So in the skeletal muscle fibers, this triad system, how it is formed in the skeletal muscle system? It is formed by one T-tubule. Okay, one T-tubule flanked by two terminal cisternae of sarcoplasmic reticulum. Okay, so as we have discussed in the previous classes, the endoplasmic reticulum, if it is present in the muscle cell, we can call them as the sarcoplasmic reticulum. Okay, so the sarcoplasmic reticulum, in the sense, endoplasmic reticulum, and the structure of endoplasmic reticulum means it is a group of cisternae. Okay, so and here also the triad system is formed by T-tubule plus two terminal cisternae. Together it is 3. So that is what triad system is. Okay, why the triad system has to be formed in the muscle cells, that is specifically the skeletal muscle fibers? So this triad system is the one helps in the rapid transmission of the impulses for muscle contraction. Okay, so this is involving in the rapid muscle transmission of impulse for the muscle contraction. For muscle contraction, this triad system is very important. Now what is the difference between actin and myosin? So there is an actin and myosin. So you should know there is a thin filament and the thick filament. Okay, so the thin filament is the actin and the thick filament is myosin. Okay, so this actin is made up of F actin. Okay, again F actin is also a polymer, and this F actin is made up of many G actin. Okay, so actin is made up of 2 F actin, and this one F and another one F, this is made up of many G actin, and this F is also made up of many G actin. F stands for filamentous actin and G stand for globular actin. Okay, so you should know actin is made up of actin is a dimer of F actin, which dimer means two filamentous F actin, together we are calling them as an actin. And each F actin is made up of many G actin, and you should know G actin will be the monomer. And this actin is made up of F actin, tropomyosin and troponin. Okay, and in the actin, tropomyosin and troponin will be bind there. Okay, so the protein and the myosin protein will be present as the structure of myosin will be, it has head and the tail region. Okay, and what will be the function of actin and what will be the function of myosin? The actin binds myosin during contraction. During contraction, the actin is going to bind with the the myosin will bind with the actin. This results in the contraction. Now here this myosin, what will be the function of myosin? For muscle contraction, there is a need of energy. Okay, so the energy will be given by ATP hydrolysis. Okay, so for the process of ATP hydrolysis, definitely there will be need of this ATPase enzyme. So only then this ATPase is the one involving in breaking the ATP to ADP plus PA. Okay, so that ADP, that phosphate group is used as an energy by the myosin, and it will be bind with the actin. Okay, so after that it needs energy and it results in pulling of the actin. It results in the muscle contraction. Okay, so this is the one pulling the actin. For pulling the actin, it needs an energy. Okay, so that will be in the form of ATP. So to ATP has to be undergo hydrolysis, that will be mediated by this ATPase enzyme. Okay, now what will be the difference between the red muscle fibers and white muscle fibers? The red muscle fibers and white muscle fibers, so based on the texture, we can categorize the muscle fibers in two. Okay, what will be the myoglobin content? So if the hemoglobin present in the muscle cells, we can call them as a myoglobin, and the myoglobin content will be higher in the red muscle cells and it will be lower in the white muscle fibers. Okay, mitochondria will be numerous in the red muscle fibers. Okay, mitochondria will be numerous in the red muscle fibers and it will be fewer in the white muscle fibers. Okay, the mitochondria will be very few in the white muscle fibers. Okay, the contraction speed, will be slow but sustained in the red muscle fibers. It will be slow and steady in case of red muscle fibers. In white muscle fibers, this contraction is very fast, so that it will make use of all the oxygen and energy instantly, and it will fatigue very quickly. The muscle will undergo muscle fatigue very quickly. It is not that much steady like as this red muscle fibers. Okay, and the example for the red muscle fibers will be postural muscles. Our posture will be determined by this red muscle fibers, and our eye muscles will be eye muscles are having the white muscle fibers. Okay, so this will be the difference between the red muscle fiber and the white muscle fiber. Okay, now name the keystone bone of the human cranium where it is located. See, keystone bone of the human cranium. Okay, that will be the sphenoid bone. Sphenoid bone, that will be the very important bone that makes the cranium, that is the skull region, cranium. Okay, so where it is located? It is located at the base of the skull, connecting with all other cranial bones. Okay, so the most important bone in the human cranium will be the sphenoid bone, and this sphenoid bone, it is present at the base of the skull, and this is the sphenoid. This sphenoid bone is the one connecting with all other cranial bones. So this is why the sphenoid bone is very important. Now, human skull is described as a dicondylic skull. we know that human skull is described as a dichondric skull. What will be the reason why we are calling this particular human skull as dichondric skull? Because this human skull is dichondric because it has two occipital condyles that articulate with the atlas vertebra, allowing the nodding movements. Okay, see this human skull, it has two occipital condyles. So this occipital condyles makes the skull articulate with the atlas vertebra. Okay, this results in the movements, nodding movements, like shaking the head. Okay, nodding movements. So this nodding movements is because of these two occipital condyles. As it is two occipital condyles, we are calling that human skull as a dichondric skull. Okay, this is the reason. And name the ear ossicles that and their evolutionary origin in human beings. So we are having three different types of ear ossicles in human body. That is malleus, incus and stapes. Okay, malleus is from the articular bone and incus from the quadrate bone and stapes from the hyomandibular cartilage. So this will be the two important things. See here, these are all the ear ossicles, and where all these ear ossicles has been evolved from, that will be the answer is from articulate bone, from quadrate bone, from the hyomandibular cartilage. So this will be the proper answer for this question. Now, so that's all about the shorts, very shorts in the musculoskeletal system. Now we are going to discuss about the very shorts in the neural control and coordination. Okay, so now the first question will be, name the cranial meninges covering the brain of man. Okay, so in human brain we are having three cranial meninges. Okay, so meninges means that is the three covering which is covering the brain. So that is the outermost membrane will be the dura meter, and the middle membrane is called as the arachnoid meter, and the innermost covering is called as a pyometer. Okay, so these are all the three coverings which is covering the brain. These three coverings together we are calling them as a meninges. Okay, so that is what outer membrane dura meter, arachnoid matter that is in the middle membrane, and the innermost layer is called as the pyometer. So all these things together we are calling them as the meninges. Okay, now what is corpus callosum? So this corpus callosum, it is a thick band of nerve fibers. Again, this corpus callosum is rich in the nerve fibers. So what is the function of this corpus callosum? This is the one connecting two cerebral hemispheres. We are having two lobes of hemispheres, left and right hemispheres. So these two hemispheres will be connected by this corpus callosum. Okay, so this as these two cerebral hemispheres has been easily connected and communicated with the help of this corpus callosum. Okay, so this is an important thing. And the third question will be, what do you know about the arbor vitae? Okay, arbor vitae is the tree-like white matter found in the cerebellum. Okay, cerebrum is a big brain, and the cerebellum is considered as a smaller portion of the brain. So in the cerebellum there is an important structure that is called as the arbor vitae, and this arbor vitae is the tree-like white matter. Okay, tree-like white matter. And what is the function of this arbor vitae in the sense? So this is the one responsible for coordinating voluntary movements and balance. Okay, so if I want to if I want to keep my hand like this, I can move. Okay, and if I want to keep like this, I can move. Okay, so this is what voluntary movements according to our consciousness. Okay, so the voluntary movement and balance has been regulated by this arbor vitae that is present in the cerebellum. Okay, now why the sympathetic division is called as thoracolumbar division? Okay, see here, there is a sympathetic division. Okay, it is called as a thoracolumbar because this nerve fibers arise from the thoracic and lumbar regions of the spinal cord. Because no doubt about that. This no critical explanation in this question. Why it is called as a thoracolumbar division means because it is originated from that division. Exactly. T1 to L2. T means thoracic and L means lumbar. So as it is originating from this thing, this spinal cord region, we are calling this particular thing as this particular division, sympathetic division, as thoracolumbar division. Okay. Now, why the parasympathetic division is called craniosacral division? Because it has a nerve fibers that will be originating from the brain. So that is why cranial nerves, and it are also the sacral region of the spinal cord. So that is why we are calling this parasympathetic division is called as craniosacral division, and the sympathetic division is called as the thoracolumbar division. Okay, so no, that much explanation is needed for that question. And what is all or none principle? And this is very important question. Okay, see here, it states that a nerve or a muscle fiber responds completely. Okay, if there is a threshold level, okay, so there is a threshold level. If the stimulus is not reaching the threshold level, there will be no reply. Now there will be no response. Okay, there is a threshold level. If the stimulus is beyond the threshold level, then the response will be very higher. Okay, either the response will be very more, or there will be no response at all. Okay, so that is what all or none principle. Okay, so it states that a nerve or muscle fiber responds completely or not at all to a stimulus once the threshold is crossed. Okay, so this is what the proper definition for the all or none principle, right? Now, how do the rods and cones of human eye differ from each other chemically as well as functionally? See here, there is a rods and cones which is present in the retina of the eye. Okay, so in the rod cells, there is a pigment which is called as a rhodopsin, and the pigment which is present in the cone cells is called as an iodopsin. Very important. Okay, so function as night vision during the night. So if the protein which is responsible for differentiating the cells which is responsible in differentiating black and white color is the rod cells. And if the cells are involving in distinguishing the colors, that is the visible region, violet, indigo, so all these red, blue, green, all these colors can be differentiated by the cone cells. Okay, now, so that is what the night vision and the dim light, the rod cells are getting activated, and in the day vision and the color detection, the cone cells are getting activated. So this will be the function of rod cells and the cone cells, and the sensitivity of the rod cells will be very high, and the sensitivity of the cone cells are very low. Okay, and it is present, the rod cells will be present in the peripheral retina, which means the outer area. Okay, outer area, the cortex region, and in the cones it is present in the central retina, and the central retina is considered as a fovea. Okay, so these are all the main important difference between the rod cells and the cone cells. Okay, now, what is mean by this blind spot and yellow spot? Okay, see where the blind spot is located, where the optic nerve exits the retina. So there is a retina. So this is the area where the optic nerve get out of the retina. So this is the region we are calling them as a blind spot. Okay, where no vision has taking place. Okay, and this yellow spot, it is also called as a macula lutea, and this is the region, this is the near the center of the retina. Okay, so in the center of the retina, that region is called as an yellow spot, and in the blind spot where the optic nerve exits the retina, that place is called as a blind spot. Okay, and the photoreceptors, very important, in the blind spot there will be no photoreceptors present in the blind spot. As there will be no photoreceptors, there will be no receptors are present to receive the light, as there will be and there will be no vision. Okay, so that is why we are calling that spot as a blind spot. Okay, and the yellow spot is densely packed cones. Okay, here it is densely packed cones. And the blind spot vision here, it will be no image formation. As there will be no image has been formed in this spot, we are calling that spot as a blind spot. And if there will be a sharpest vision in the yellow spot, that is why we are calling them as the yellow spot, that is macula lutea. This is what the difference between this blind spot and the yellow spot. Okay, Now, what is organ of Corti? So, this organ of Corti is the sensory structure of hearing located in the cochlear duct of the inner ear. It contains the hair cells that convert sound vibrations into nerve impulses. Okay. See, you should know this organ of Corti is a sensory structure. Okay. Sensory structure, which means in the ear. This is a part of the ear. In the ear we are having a cochlear region. Okay, cochlea. So there is a cochlear duct which is present in the inner ear. We are having outer ear, external ear. That is what we are calling them as a pinna. Okay, so there is a middle ear and there is an inner ear. In the inner ear we are having a cochlear duct. In the cochlear duct we are having a structure which is called as an organ of Corti. Okay, so in the organ of Corti, we are having a hair cells. So this hair cells which is present in the inner ear, this is the one involving in converting the sound vibrations into nerve impulses. This is very, very important. Okay. Now, so that's all about the very shorts in the neural control and coordination. Now come to the endocrine system and chemical coordination. Okay. So the first question will be, what is acromegaly and name the hormone responsible for this disorder. So the acromegaly is a disorder caused by the excess secretion of growth hormone. See, there is a growth hormone. Okay, there is a growth hormone. If the growth hormone secretion is higher, it leads to gigantism. If it is lower, this leads to dwarfism. Okay, again if it is higher in adults, in case of adults, that condition is called as an acromegaly. Okay, so in case of, see, this acromegaly is a disorder. Okay, why it is considered as a disorder? Because there will be the hormonal dysfunction. And what will be the name of the hormone? That will be the growth hormone. Okay, so growth hormone, that too specifically it is in the adults. This leads to the abnormal enlargement of hands, feet and facial bones. Okay, so this condition is called as an acromegaly. So you should know what exactly the term acromegaly, which hormone is responsible for, and what will be the symptoms. Okay, now which hormone is called antidiuretic hormone? Write the name of the gland that secretes it. See here. Okay, see, we are having a pituitary gland. Okay, so and we are having the anterior pituitary and posterior pituitary. So in the anterior pituitary, we are having growth hormone, thyroid stimulating hormone, follicle stimulating hormone, luteinizing hormone, prolactin. All these hormones were synthesized by the anterior pituitary. And in the posterior pituitary, so this we are ADH, that is the antidiuretic hormone, and this antidiuretic hormone is also called as a vasopressin. Okay, and also this posterior pituitary is also having another hormone which is called as an oxytocin, and you should know it is not secreted by the posterior lobe of the pituitary gland. Okay, but instead it is secreted by the hypothalamus. It is secreted by the hypothalamus, and the only thing done by the posterior pituitary, it is just storing it. Okay, so the antidiuretic hormone is synthesized by the hypothalamus and it will be stored in the posterior lobe of the pituitary. Okay, so this is the proper answer. And the difference between diabetes insipidus and diabetes mellitus. Okay, see, the diabetes insipidus means deficiency of antidiuretic hormone. See, what is meant by antidiuresis process? So the kidney is the one involving in removal of waste. Okay, so in the form of urine. So now this kidney has to be eliminate the water to for up to the toxins has to be eliminated. Okay, so toxins can be easily eliminated out from the body only with the help of water, and the water is not needed excessive in an excessive amount. What are all the waste to how much amount of waste has eliminated in the body, that much amount of water is sufficient. Okay, that much amount of water is has to be eliminated out from the body, but that the water reserve, so other remaining water has to be reabsorbed by the kidney. Okay, now who is responsible for is a resorption of this water? So that is what this antidiuretic hormone. Okay, so if this antidiuretic hormone is not functioning properly, more amount of water has been eliminating out from the body. So this if the deficiency of ADH, this results in excessive dilute urine. Excessive dilute means if water is more in the urine, we are calling them as a dilute urine. If water is very less, we are saying that as a concentrated urine. Okay, and here the blood sugar level is very normal. And in the diabetes mellitus, here the deficiency or resistance to insulin. Okay, the insulin secretion, the hormone insulin is not functioning properly, so that results in the condition called diabetes mellitus. So here in the urine, lots of glucose has been eliminated, and here the blood sugar level is also higher. Okay, so see here in the blood, the excessive glucose has to be converted into glycogen, and this conversion has been done only with the help of the insulin hormone. If insulin is not if the insulin is not properly present in the properly if insulin is not functioning properly, the excess glucose which is present in the blood, which is not converted into the glycogen, so that will be so we are saying that the blood glucose level is higher if the insulin is not functioning properly. This results in the condition called diabetes mellitus. Okay, and the glucose will also be eliminated in the urine, so that is why we are saying excessive glucose in the urine, and the blood sugar level is also higher. Okay, next question, what are islets of Langerhans? See, the islets of Langerhans are the clusters of endocrine cells in the pancreas. Okay, so they contain alpha cells. Alpha cells. See here, we are having a pancreas. In the pancreas, we are having alpha cells, beta cells, delta cells, and epsilon cells is also there. Okay, see, the alpha cells is the one responsible for secreting the hormone called glucagon, and the beta cells are responsible for secreting the hormone called insulin. Okay, see, this islets of Langerhans is a cluster of endocrine glands which is present in the pancreas. Now they are containing the alpha cell, beta cell. So the alpha cell is the one responsible for secreting glucagon, and the beta cell is secreting the insulin. So this is the one regulating the blood sugar levels. Okay, just now we have seen. And what is insulin shock? So insulin shock is a condition. It is caused by an excess insulin. This leads to hypoglycemia. Okay, see here, there is an insulin. So what I have mentioned you before, the excess glucose in the blood which has to be converted into glycogen, and it has to stored in the liver and the muscle. Again, whenever the body is in need of glucose, this glycogen is once again converted into glucose with the help of the glucagon hormone. Okay, now the insulin shock is a condition where there is an excessive synthesis of an insulin. This leads to low blood sugar level in the blood. Okay, so that condition is technically called as hypoglycemic condition. So this may result in unconsciousness or coma. So this is called as an insulin shock. Okay, now, so which hormone is commonly known as fight and flight hormone? So we are having an adrenaline hormone which is also called as an emergency hormone. So why we are calling this particular hormone as an emergency hormone means, so this hormone will be secreted only in the time of emergency. So that is why we are also called this adrenaline hormone as epinephrine hormone, or we can say flight and flight hormone. This is secreted by the adrenal medulla. So we are having adrenal cortex region and adrenal medullary region. Adrenal cortex is responsible for secreting the glucocorticoids and mineralocorticoids, all these things. Adrenal medulla is responsible for synthesizing the adrenaline hormone, that is epinephrine hormone. Okay. Now, what are androgens? Which cells secrete them? Okay. So androgens is called as the male sex hormones. Okay, so the male sex hormones are nothing but testosterone. They are responsible for male characteristics. Okay, so androgens is called as a male sex hormones, and the male sex hormones is like the testosterone. This is the one responsible for male characteristic features, male sexual characteristic features. Okay, now, so how this male sex hormones has been secreted, like testosterone? It will be secreted by the Leydig cells. Leydig cells will be present in the testis region. Okay. Now, What is erythropoietin and what is its function? So erythropoietin is very important. It is a hormone secreted by the kidney. Okay, and why the kidney has to secrete the hormone called erythropoietin? Because this erythropoietin is involving in stimulating the red cell production in the bone marrow. Okay. So in the bone marrow, hematopoiesis process will be taking place. Hematopoiesis means production of blood cells. Okay, so now this production of blood cells, and the blood cells are RBC, WBC and platelets. Now this erythropoietin is responsible for stimulating the red blood cell production in the bone marrow. Okay, so this is what the importance of this erythropoietin. Okay, so that's all about today's class. We'll meet you in tomorrow's class with another chapter, short answer, very shorts. Okay, thank you.